Good day everyone, and Bu Retno, thank you for the time and the opportunity to checking my video. Today I will be presenting to you my study under the subject for hematopoietic stem cells for the preclinical study. My name is Jennifer Monica, and my presentation today will be covering the paper from the Colella and team that published on 2024 under the title CNS-wide repopulation by hematopoietic derived microglia-like cells correct progranulin deficiency in mice. So basically they do the transplantation of the HSCs and then to do the repopulation of the CNS or central nervous systems. My presentation today will be covering all of this from the introduction and then the methods and then the outcomes and the conclusion. First, for the hematopoietic stem cell, for introduction, are the stem cells that will be blood and immune cells. They can get it from the bone marrow like this. They have the needs for the stem cells. Then we can get the HSCs. So after the transplantation of the HSCs, we hope they will rise into the blood and immune cells. And then we can get it from the bone marrow transplantation, like from this paper. A subset will migrate from the donor, and then the HSC derived cells will reach the CNS, and then they differentiate into the microglia-like cells. But the microglia engraftment is limited still, and this restricts the therapeutic use for the neurodegenerative or lysosomal diseases. So interesting enough, we will need to improve the CNS entry and the differentiation of the donor. So not only we have to inject them, and we need to make sure that they can reside stay in the targeted area. So this paper give rise for the problem statement, which are the main problem is there is already a standard myoablation. They use the busulfan, but the microglia replacement still very poor. So without the adequate CNS engraftment, the transplanted HSC can deliver therapeutic proteins. So we need to open the CNS niche safely and efficiently for donor-derived cells. So we need we need to make a room for the donor to reside and help the recipient. So this paper have this research aim, which is to test whether an optimized conditioning regimen of busulfan plus CSF1R inhibitor PLX3397 will enable HSC-derived microglia-like cells to robustly repopulate the CNS and correct progranulin deficiency in mice. So the study design overview, they use the InfoHome model. So there are the adult homozygous C57BL6 that already tagged with the GFP mice. They use it for the bone marrow donor, so they can track it the cells from the donor in the recipient. They use the recipient mice. The model that already been knocked out for the GRN gene, so they have deficiency in progranulin. So, and then the adult mice that is for the recipient, they do the conditioning using the busulfan for the myoablative, and then they add the PLX3397 by oral 100 milligram per kilogram per day from the day 15 until the day 20. So this is to aim to do the depletion microglia temporarily, and then they do the transplantation for the donor bone marrow HSC. They have the tag already with the GFP, green fluorescence protein. They injected it using the IV administration route, and then they assess the engraftment using the GFP tracking, and then they check the single cell RNA sequencing, and they check also the lipofuscin and the protein. So basically there are three key methodological innovation that they bring up in this paper. First, they do the myoablation with the busulfan, which is already a standard for it, and then they also do the glia depletion, the CSF1R blockade. So this will hopefully depletes the microglia from the recipient, the original one, to open the CNS niche so the donor can come and occupy that area. And then they also do the high resolution profiling. They check the single cell RNA sequencing to show the lineage identity of the donor derived cells. And for the third one, they check the therapeutic test on GRN mice, the knockout mice, for the neurodegeneration models. And then we come to the outcome metrics. They check the molecular restoration. First, for the result for the engraftment efficiency, they reported that the busulfan and the PLX give more than 90% CNS microglia replaced by donor-derived GFP plus cells. So this is just to confirm that when they add the PLX, the inhibitor, they have the way higher microglia from the donor compared to the busulfan alone. So the replacement is very low still with the standard old method. And then they also reported that the engraftment is stable for more than 6 months. They observed it in multiple brain regions, including the cortex and the retina. So this is the image for the report for the immunofluorescence. We can see that the GFP tags are showing in widespread across the brain region. I mentioned here, I wrote here, in the busulfan and the PLX versus the busulfan alone, we can see the comparison that this intervention is giving higher signals compared to the busulfan alone. So this meaning we can interpret that the combination of the myeloablation with the busulfan and the short CSF1R inhibitor treatment, which is the PLX3397, is dramatically accelerates and increased replacement of host microglia by donor-derived MGLCs, the cells that differentiate from the bone donor. This could happen by two mechanisms. First is they check from the cytokine and the chemokine search. So we can see that there's a temporary search in all of these. There is a serial cytokine analysis performed on whole brain lesions by 48 plex luminex, and they can see on basically until the day 24 there's a higher signals reported. So we can conclude from this graphic that the temporary cytokine and chemokine search means they promote migration and integration. Basically, they to put it in a simple way, they calling other cells to come, calling the other donor cells to come and resides in the targeted area. So, and then this is the second mechanism. They check, this is the result from the single cell RNA sequencings. We can see that the donor-derived microglia-like cells show heterogeneous transcriptome. So this means there's a homeostatic microglia, and also there's this gene that I put in a green box are the embryonic microglia genes. So this is suggests both niche availability and chemoattraction drive the engraftment. And then they also check the efficiency for the therapeutic function. We can see here they compare the wild type and then the KO models, which the knockout of the GRN mice that have brain pathology. We can see the lipofunction, the blue, build up and no progranulin, compared to the myeloablation and then myeloablation with the microglia inhibitor. We can see here also that this is the engraft efficiently done, and then from the ELISA and the Western blot, they can see that the progranulin levels are restoring. So this is higher for the intervention with the busulfan and the PLX compared to the busulfan alone. In these two, we can see that they reduce neurodegenerative changes for the lipofuscin. That is the marker for the brain pathology. This we can interpret this as donor-derived microglia cells can restore the progranulin or PGRN levels and reduce pathology in GRN knockout mice. And then this is just showing the improvement that we can see here. The intervention that they propose can restore compared to the knockout one, even though it's not as high as the wild type. This is also explaining the same thing. So basically the busulfan with the PRX3397 that inhibitor reconstitutes progranulin in the eye and improve lipid metabolism and proteostasis in the brain of a mouse model of CLN11 FTD. So this is another assay, further assay that they do to check the therapeutic function. So the significance of the translational impact, we can see that before they not only they have the proof of the HSC transplantation is delivered and then reside in the targeted area, but they also have the function. They have higher translational value because the function of the therapeutic is proven already. So the busulfan with the CSF1R inhibitor regimen uses the drugs that are already known in clinical settings, so it's more easily to translate into the human research. This also as a framework for the future HSC-based gene cell therapies for the neurodegenerative disease. But however, there is still limitation or a critical discussion that we can discuss. First, the donor-derived microglia is not identical to the native microglia, so there is still a functional equivalence in uncertain. Maybe we need to check. We need to have further research to check that. And then also the long-term durability and the safety in humans are still untested. Also, the conditioning toxicity may limit the clinical use. The drug that they use there. Maybe we can explore other option. Also, the partial restoration, which means will need higher expression or multi-cell targetings. Also, this is still in a preclinical study, so there is a species differences. The mouse CNS more permeable than the human, so maybe they will need adjustment for that. So we come to the conclusion. First, the busulfan and the CSF1R inhibitor enables extensive CNS microglia replacement by the donor HSC progeny, and then demonstrated the therapeutic correction of progranulin deficiency in mice. So not only they transplanted into the mice, but they also have the function proven. And then they represent a major preclinical step forward the HSC-based therapies for the CNS disorder. So for the future research, we can explore the optimizing doses and then the safety and then gene-modified HSCs for the human translation. That's all for my presentation today. Thank you for your time and thank you. I think that's all from me. I hope this presentation can illustrate the function of the hematopoietic stem cells for the preclinical study, and if there's any review or a comment, you can contact me. Thank you very much for your time and attention. I hope you have a good day.